Hi, I'm Ari. This is my project on extracting antibacterial compounds from soil bacteria. So for this project, we searched for antibiotic activity in microbes that we collected from soil samples around St. Olaf campus. We did this through a program called Tiny Earth. They promote and fund ongoing research for antibiotic discovery. This is important because antibiotic resistance is a global issue currently where many pathogens have developed resistance to antibiotics we throw at them. This can have dangerous consequences, including superbugs and cases where previously treatable infections become life-threatening again. In this case, we found an isolate collected on the edge of Big Pond. I called it RR4. We tested it against escape relative pathogens. So these are safer versions of the most dangerous and common pathogens. And we found that it produces a zone of inhibition against Staphylococcus aureus, Staph epidermidis, and some effect against E. coli, although the results were a little unclear. So now that I knew this strain had this desired trait, I wanted to learn more about the bacteria. So I ran a series of biochemical tests to characterize RR4. This included a gram stain test, where I saw that it was gram positive. You can see this microscope image here. I also saw that it was rod-shaped. Through a DIC microscope, you can see that the bacteria were motile. Through a catalase test, we observed that it was aerobically respiring. I also saw that it was sensitive to several antibiotics, including streptomycin and penicillin. After that, we also performed PCR and amplified its 16S ribosomal gene for sequencing. After receiving the actual string of base pairs, I ran it through a BLAST software that compares the sequence to others in the database. The closest match was a bacteria called Bacillus pumilus. Bacillus pumilus is a very interesting bacteria. It's a very broad-range species, meaning many bacteria often get classified as Bacillus pumilus. It has also been researched for its antimicrobial properties. Several antifungal agents and antibiotics have been discovered from Bacillus pumilus. Here's a phylogenetic tree where you can see RR4 is closely related to Bacillus pumilus, as well as several other Bacillus species. These are found in space in the outer stratosphere, which I thought was pretty interesting. These names are of other students who had closely related species. Finally, my third kind of discovery was that these compounds can be extracted through a process of freezing and lysing the cells with ethyl acetate. These compounds can then inhibit the pathogen by themselves, seen here by this zone of inhibition around this disc diffusion. I also tested the extract against yeast. It did not show a zone of inhibition. This is good, however, because yeast is a eukaryotic cell, and so it's much more closely related to us than the bacteria. So this could be evidence that RR4 and its extract are good candidates for further antibiotic discovery. For more information on Bacillus pumilus, you can use this QR code in the bottom right. That's all. Thank you.